Hi friends, I am Varsha Gaitonde and welcome to my channel. This topic is about the Nobel Prizes related to genetics and plant breeding. We'll see one by one. In 1933, first time Thomas Hunt Morgan, who got Nobel for Drosophila genetics, including sex linkage and development of theory of gene, that is linkage theory. In 1935, Spearman he studied on embryonic induction. In 1946, Muller got Nobel Prize for the discovery of mutagenic action of X-ray in Drosophila melanogaster. In the same year, Sumner got in the field of chemistry for crystallization of enzymes. Stanley got for purification and chemical characterization of viruses. In 1952, Martin and Sinje got Nobel for chromatographic techniques. In 1952, Salman Waxman for the discovery of streptomycin, the first antibiotic effective against tuberculosis. In 1957, Todd got for the studies on structures of nucleosides and nucleotides. In 1958, Beadle, Tatum and Lederberg got Nobel for Neurospora biochemical genetics and their studies on bacterial conjugation. In 1958, Sanger got Nobel for studies on protein chemistry. In 1959, Ochoa-Korenberg for in vitro synthesis of DNA. In 1962, Watson, Crick and Wilson got Nobel for DNA double helix. In 1965, Jacob, Monod and Loaf got Nobel for operon concept of gene regulation, means Jacob and Monod got for operon concept and Loaf got for microbial genetics. In 1966, Ross got for studies on oncogenic viruses. In 1968, Nirenberg, Hargovind Khorana got for their deciphering genetic code and along with Hawley for in vitro synthesis of gene or DNA. In 1969, Delbruck, Luria and Hershey got Nobel for their contribution to viral genetics. In 1970, Borlaug, who is also called as father of Green Revolution, got Nobel in the field of peas for the development of semi-dwarf wheat varieties. In 1974, Claude, Dewey and Pellade got for the contribution to cell biology. In 1975, Delbruck, Tamin and Baltimore got Nobel for the studies on oncogenic virus, that is for reverse transcription. The process of reverse transcription is also known as Taminism. In 1978, Smith, Needhams and Erber got Nobel for their utility of restriction endonuclease for the study of genetic organization. In 1980, Berg, Gilbert and Sanger in chemistry got Nobel for the experimental manipulation of DNA, including production of rDNA, that is recombinant DNA, or we may say that is in the field of genetic engineering. In 1983, McClintock got Nobel for the discovery of transposable elements, even though McClintock studied or did her experiments in 1950s, but she was recognized after a long period, that is almost 30 years. In 1984, Jern, Kohler and Milstein got Nobel for immunology, including hybridoma technology. In 1986, Ruska, who got Nobel for designing the first electron microscope. As you know, Knoll and Ruska are the pioneers for discovering the electron microscope. Then in 1989, second Altman got Nobel for ribozyme, that is a game-changing enzyme in the field of RNA. In 1990, Murray and Thomas, they got Nobel for organ and cell transplantation. In 1993, Smith got Nobel in chemistry for fundamental contribution to the establishment of oligonucleotide-based site-directed mutagenesis and its development in protein studies. In 1993, Sharp and Robert got Nobel Prize for split genes. In 1997, Prusiner got Nobel for the discovery of prions. Prions are a new biological principles of infection. These are containing only proteins. In 2002, Sidney Brenner, Harvis, and Sulston got Nobel for the discoveries concerning genetic regulation of organ development and programmed cell death. This you need to remember in 2002, Brenner, Horwitz and Sulston got Nobel for the programmed cell death process. 2006, Andrew Fire and Craig Mello in the field of medicine, they got Nobel to decipher how RNA interference can switch genes on and off. In the same year, Korenberg got Nobel in the field of chemistry for the study of molecular basis of eukaryotic transcription. In 2009, Blackburn, Grieder and Zostak in the field of medicine got Nobel for discovering how chromosomes are protected by telomere and telomerase enzyme. In the same year, in the field of chemistry, Venkataraman Ramakrishnan, who is an Indian, got Nobel for the studies of structure and functions of ribosomes. In 2014, Eric Hall, Stephen Moreover and Williams got Nobel for development of supersolid fluorescence microscopy. In 2015, Thomas, Paul and Aziz got Nobel for DNA repair mechanism, that is nothing but mechanistic studies of DNA repair. In 2007, Jacques, Frank, Joaquin and Richard got Nobel for the development of cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution. If some scientists are left over here, please comment in or put a comment in the comment box so that I will also know it and I can also tell to other people. Thank you.